All right, welcome everybody. Welcome to our second chair's lecture this year. It's my pleasure to introduce today's speaker, Warren Wakarchuk. Did I get that right?
That's good.
Okay, right, good. Warren is one of our newest recruits. He's a professor who started with us in May. He's come to us from Ryerson University, where he was a chair from 2016 to 2019, I guess. And Warren comes here as currently the associate scientific director of, which I know is GlycoNet, but it's also the Canadian Glycomics Network. Are they
Correct.
the same thing?
Yes.
Or those are just two names for the same thing?
Two names for the same thing.
Okay, right. Which is, of course, as we all know, is a big enterprise here on campus. And we think that Warren will be moving into a leadership position there over the next year, segueing into that. A little bit of history about Warren. So Warren was at Ryerson since 2012, and before taking a faculty position at Ryerson, he spent close to 20 years, I think, in the National Research Council in Ottawa, where he's been a practicing glycobiologist. I probably should have said that at the top. That's what he is, what he does. Is that fair enough?
Fair enough.
Okay, fair enough. Where he did glycobiology with the NRC. Did a lot of work with industry as well. So we're going to hear some of what Warren has done in his career today. His glycobiology talk, carbohydrates are not just for breakfast anymore. And to go back a little bit further, Warren is a Canadian. Warren did most of his graduate and undergraduate work at UBC. Correct me if I'm right about that.
Yep.
And you finished high school in Medicine Hat. Correct. But you're not an Albertan, you're a Saskatchewan,
Yes.
right? So he's just like a lot of other Albertans. He's not from Alberta, but he's come back.
Indeed.
Or he's an Albertan who's, okay. I lost my thread there. He's not really an Albertan, he's from Saskatchewan, but he's come back. He's come from away.
Come from away.
That's what I'm looking for. He's come from
Indeed.
away, which is true for many of us here. Okay, with that, I'm going to stop yammering and give you Warren.
All right. Thanks, Dave. All right. It's a real, yeah, lights are great. It's a real pleasure to be able to talk to my new colleagues about the work that's going on in my lab. And I structured this talk in two parts. One is kind of an introduction, which is more about why you should care about glycobiology, how could it influence day-to-day life for you and its place in biology. So the reason that you should care about it is every cell surface, doesn't matter whether you're a lowly microbe or us, all your cells are covered with carbohydrates. And so the carbohydrates play the role of the first responder in biology, because if you look at the glycocalyx on a red blood cell, this particular layer is mostly carbohydrate stuck on the ends of proteins. And anything that needs to encounter that cell has to get through that forest of glycans. And so it's a formidable barrier, but it also plays an important role in signal transduction and reception of traveling molecules that are circulating around. And the same is true, of course, for bacteria. Bacterial capsules are the thing that prevent them from being phagocytosed, very important virulence factor. Fungi, of course, doing that as well. And then plants are exquisitely good at glycobiology. They encapsulate themselves in carbohydrate. Their cell wall is very heavily carbohydrate influenced. And so everybody is in on this glycan or glycoconjugate game. So glycans actually represent kind of a cool version of biodiversity. So not all glycans are the same. We have very similar building blocks, but they're used in very different ways. And so glycobiologists have come up with this particular color scheme to get rid of the complicated chemical structures that make these structures fill up 2 or 3 pages worth of space in order to be able to represent them in a reasonable way. And so if we look at viruses, envelope viruses from mammals steal glycans from the host membrane. So these glycans look a lot like vertebrate glycans. But the bacterial ones, the yeast ones, the fungi and plant and nematodes all look very different. We have the same machinery, same kinds of enzymes, same kinds of precursors, but you can sort of speciate things on the basis of the kind of glycans they have. And so they're a good indicator of biodiversity and a good reason why, when you're working on model organisms, for example, some of the interactions that you're looking at don't transfer to humans particularly well. And that's particularly important for things that go on in the brain, because our brain glycobiology is completely different from many other organisms. All right, so glycans mediate a host of interactions at the cell surface, as I said, and I'm not going to go through this slide in great detail, but there's a few things that are absolutely critical. So cellular interaction signaling between cells is absolutely requires glycans. Neural network building, irregardless of what neural tissue you're talking about or what organism, requires glycans at some level. Wound repair, cell migration, nutrient sensing, response to growth factors, response to pathogens, etc. All of that stuff is dependent on cell surface carbohydrate interactions at some level. And then if you want to get into therapeutic avenues, if you want to use an antibody to kill a particular infected cell, for example, that antibody-dependent cytotoxicity requires that the antibody have a glycan on it. And that glycan has to have a particular structure in order for it to work properly in that reaction. Otherwise, it's not recognized by the receptor. And so that's absolutely required. And then I'm going to tell you today a little bit about protein-based drugs. But before we get to protein-based drugs, I thought it would be good just to review why or how carbohydrates contain so much information. So these are two very common sugars. So we've got table sugar, sucrose here, and we've got milk sugar, lactose here. And oops, let's go back here. If we take a look at the 3D structure of these things, these two sugars have exactly the same empirical formula, but they have totally different 3D space. So the reason that sucrose is sweet is because the receptor in your mouth recognizes it. And the reason that lactose tastes a little bit like sand is because it doesn't fit into the sweet receptor. And so there's no way for it to be recognized as sweet. And so I think this is a really simple idea that you can think about. You know, how is receptor recognition of a carbohydrate involved in a particular interaction? So if you're lactose, you're not perceived as being sweet, so the receptor doesn't trigger, but sucrose triggers very nicely. So I tend to think of carbohydrates then as very information-rich molecules because they have this wonderful 3D space that they can rotate around in. Carbohydrates are involved in much of the everyday biology that everybody knows about. And those of us who have been working in glycobiology for some time see the potential for new medicines coming out of this study. So let's take another look at an everyday occurrence. So we think about ABO blood groups a lot. And I think everybody's familiar with the ABO blood group system, but you might not realize that the molecule that determines your blood type is actually a carbohydrate. That carbohydrate sits on the surface of your red blood cell, and it determines whether or not you will be able to go through a compatible blood transfusion. So our immune system then is primed to whatever molecule is there for the ABO system. So if you've got an A antigen, you can't take B blood. If you have B, you can't take A. Everybody can take O, and we can do that because the O individuals don't generate that immune response to A and B, and that's why O blood can be the universal donor. Okay, so bottom line, transfused red blood cells then need to have the correct antigen, and that antigen is a sugar. It's a carbohydrate structure. So what does it look like if we use that ball and stick representation that we were talking about before. So the O blood group can be simplified to having these sort of 5 base sugars. And then that's converted into the O blood group through an enzyme called the blood group A glycosyltransferase, and it transfers a square onto this circle to give you the A blood group. And then on the other side, we have the blood group B transferase, which transfers a circle onto the circle. And so the difference between blood group A and B is this one has a square, this one has a circle. They're very close in chemical structure. And the fact that the body can discriminate between these two things means that the carbohydrate is an exquisite recognition element for the immune system. What's also really cool from an enzymologist's point of view is that these two enzymes must have arose from a gene duplication at some point, because there's only 4 amino acids different between them. And of course, they're in the active site of the enzyme, which gives them the specificity for square or circle. So from a protein engineering point of view then, which is something that we've been doing, it's nice to know that you wouldn't have to change too many amino acids in order to change the specificity of an enzyme or to tweak it to do something that it wasn't naturally evolved to do. Okay, right, so just to reiterate that those two blood groups really only differ by a very tiny chemical structure. Colleague of mine in Vancouver, Steve Withers, has been working on a way of converting blood group A and B into O. And the reason for that, of course, is that the number of people who donate type O blood isn't as high as it needs to be. And so it would be really great if we could just take A and B blood, treat it with an enzyme, convert it into O blood, and carry on. And so they have, for a number of years, been screening microbiomes for bacteria that have the capability of specifically converting blood group A into O or blood group B into O. Turns out blood group B into O is pretty easy, but A into O is not. And they had a nice paper in Nature Microbiology earlier this year, which describes actually a 2-enzyme system that they came up with that can turn type A blood into type O. And so this is obviously of great interest to people in the transfusion world. And so we're hoping that that can be transformed into some kind of commercial enterprise for blood supply. All right, so I think thinking about blood group antigens, then it's a really good example of how carbohydrate structures occur in everyday biology, and they can be a matter of life and death, right? If you're transfused with the wrong blood, you could die. That's pretty clear. And the chemical structures between those two glycans are really tiny. And so these small differences then can be very significant. And what's also interesting is that the pathogens that affect us really have honed in on those carbohydrate antigens. And many of the viruses that affect us really have a tropism for a particular blood group. And those of you with blood type O are going to be more susceptible to a large number of viruses. It's just a fact. It's just a fact of life. That's unfortunate, but it's a fact of life. Okay, so when we started getting into glycobiology, we were very interested in this idea of molecular mimicry. And I think in this particular room, many of you are familiar with mimicry in biology on a different level. But it's, and the example here is the monarch versus the viceroy butterfly. And the viceroy butterfly is quite palatable to the predators that would eat it, whereas the monarch is not. And so the viceroy has come up with this idea that I should look like the monarch so that I don't get eaten. Good strategy. Bacteria have done exactly the same thing. They've said, hey, if we look at the blood group antigens, or we look at carbohydrate antigens that are on human cells, why don't we decorate ourselves with something that looks like that, and then we won't be eaten either. And so we started looking at bacterial pathogens that infect humans, And we knew that they were capable of making human-like cell surface carbohydrates. And so we came up with this analogy that bacterial pathogens were basically a wolf in sheep's clothing. And so they coated themselves in this sugar coating so that the host immune system said, yes, that is a human carbohydrate antigen, no worries, let it go. And then they relied on other virulence factors in order to cause disease. And this is a very common strategy for a lot of bacterial pathogens. So Neisseria meningitis, Neisseria gonorrhea, Haemophilus influenza, there's a big long list of these things. And they all have in common that they use sugars that look identical to the ones that are present on our cells. So the take-home message from that was then that we knew that bacteria had enzymes which could make human glycan structures. And bacterial enzymes are much easier to work with in the lab than human enzymes are, although we're getting better with the human enzymes. So what we decided to do then was look at using bacterial enzymes to synthesize carbohydrate structures that were important in biology, so bioactive carbohydrates. And we were able to do a lot of these reactions, which are very difficult chemically. They're multi-step chemically, but you can link two sugars together with one enzyme and one sugar donor. And so over the years, we've used about 24 different enzymes, and we made a whole bunch of soluble oligosaccharides that can be linked to various things, and people have applied it in a variety of ways. But we realized that even though we could make these bioactive glycans in a test tube, wouldn't it be cool if we could reprogram a cell to do that job for us by putting the biosynthetic pathway in, and thereby getting rid of the need to purify the enzymes and do all of that work. And so we set about a synthetic biology project to re-engineer E. coli in order to make protein-based therapeutic drugs. So why were we interested in therapeutic proteins? Well, in 2017, the global market for these so-called biologics and biosimilars was around $210 billion US. That's a very large market, and there are a lot of people who are on protein-based drugs for a variety of reasons. So blood factors for hemophilia, anticoagulants for surgery, antibodies are huge, so anti-cancer strategies are a very large part of the market these days. Anti-inflammatory antibodies, things like rheumatoid arthritis or Crohn's disease, for example. So there's a lot of those. Hormones, human growth hormone, the cyclist drug of choice, erythropoietin, which stimulates the production of red blood cells, probably one of the most abused proteins on the planet. It also has clinical applications, but so there's a whole bunch of these interferons, and then enzyme replacement therapies for people who have things like Gaucher's disease or other lysosomal storage diseases. So it's a huge opportunity then if we can apply what we know to doing that. And so why do we care whether these proteins have glycans or not? One reason is that in order to stay circulating in the blood, we have these liver receptors. So this particular one is called the acylo-glycoprotein receptor, and all that means is that if you had a carbohydrate that had a sugar called sialic acid on it, it wouldn't interact with that receptor and it would just keep circulating. But if that sialic acid comes off, or it didn't have a sialic acid to start with, it's captured by this receptor and it's removed from circulation. And many of the early therapeutic proteins, of course, were produced in systems that did not have authentic glycosylation, and so they had very short half-lives. So you had to be injected with a giant bolus of the drug in order for it to do anything. And then as we got smarter about keeping them in solution, we figured out that if we stuck this sialic acid sugar on the end, we could keep things in circulation much longer. And it also turns out that in certain proteins, having this intact glycan protects from proteolysis, and this is also a very important factor when things circulating in your blood. Your blood is a soup of proteins that do all kinds of things, and we have proteases and other things in there that help degrade proteins as well. So back in 2011, we did a study where we had a particular protein called alpha-1 antitrypsin, which, as its name suggests, is an inhibitor of a protease called trypsin, and it finds its function mostly in the lungs, where it inhibits elastase, which is a protease that's secreted by the white blood cells in the lung tissue. And there are a certain number of people who are born without A1AT or a defective version of it, and they have trouble with lung damage then. So neutrophils get activated in the lung to get rid of particles or other things that are occluding airways, and there's nothing to shut off the proteases that are turned on, and so then the lung gets damage from its own protease. And so this is a therapeutic protein of some interest. And what we did was we took it and we made two different glycan modifications just on the existing protein. We just took the protein as it comes from human serum, made a couple of slight changes to it, and then we actually measured how long it stays in the blood. And so this is basically just an area under the curve plot where we took normal serum-derived protein, our first modified protein, and our second modified protein. And the second modified form is cleared about 15 times slower. And what that means is that it has about a 5-fold improvement in half-life. And that's the difference between a daily injection and once a week. So it should lower the cost. It should give better patient compliance. There's a number of things that could be beneficial from having that change in protein half-life. So what's our lab doing these days? So we're looking for how to apply these enzymes to solve various problems in glycobiology. So the story that I'm going to tell you about now is reprogramming cells through synthetic biology, but we have these other programs going on as well. I told you we were able to make lots of small molecules. We have a very exciting program in tissue regeneration that's just in its infancy at the moment, and I'd love to talk about that someday. And we've been doing a lot of remodeling of existing glycans on therapeutic proteins to improve their efficacy. Okay, so what does reprogramming E. coli look like? If you look at my stylized E. coli here, what we've done is we've added genes and we've integrated them into the host genome. And these genes come from a variety of places, as I'll show you in a minute. And this then gives a new biosynthetic pathway to the bacterium. And then we take a plasmid which expresses a particular target protein that we're interested in, so in this case a therapeutic protein. And what we hope that will come out of this is designer strains of E. coli then that are tailored for making specific therapeutic proteins that have the appropriate carbohydrate on their surface. So proteins that we've been working on so far tend to be in the cytokine family, cytokine or growth factor families. So I'm going to tell you a little bit about interferon alpha-2b, which is an antiviral protein. It's also used as an anti-proliferative in cancer therapy. It has really bad side effects, though. And so patient compliance on interferon is quite bad. And we're also working on an alternative cytokine, but we're just starting that project, and hopefully I can tell you about that at some time in the future. But this is clinically used. It's used a lot. It just makes people feel bad when they have to use it. Human growth hormone, obviously used for treating stunted growth, but it's also being used to prevent muscle wasting that was associated with HIV/AIDS and other muscle wasting diseases, where it just helps to build up tissue for those patients. And then we've been working on these antibody fragments called single-chain antibodies or single-domain antibodies. So these are about 1/5 the size of a normal antibody, so they're much smaller than a regular antibody. And they have very good tissue penetration, so they're good for targeted immunotherapy, and they're very good for diagnostic imaging, but they stay in the blood for about 5 minutes. So we need some way to keep them in the blood, so obviously our trick of adding carbohydrate to them would be of benefit. And then using E. coli as a bioreactor because it's got a very short growth time, it makes lots of biomass, and it's very easy to work with. And of course, as soon as you don't have to grow anything in fetal bovine serum or some serum replacement, your costs go way, way down. Okay, so the biochemical pathway that we set out to do is called the core-1 pathway, and you don't need to worry about what core-1 means. It's just there are 7 different cores, and we just picked core-1 because it's the most common of this type, and it's what's actually found on the mucin-type proteins in your body, so it's very common. And what it involves is adding a square to a serine or threonine, and then taking that square and adding a circle, and then to the circle we add a diamond. And we can add 1 diamond or 2 to end up with this tetrasaccharide, so structure with 4 sugars, which has 2 of those sialic acids that would prevent it from being picked up by that clearance receptor in the liver. So at the same time as building in this biochemical pathway, we've been doing protein engineering because it turns out that it's fairly easy to manipulate the site on a protein where the glycan is actually added. So in the class of proteins that we've been working on, both interferon and HGH have these 4 central helices that are part of the intrinsic structure, and they're held together by disulfide bonds. And then they have this unstructured loop. And in interferon alpha 2b, there's a natural glycosylation site in that loop, so it's easy to look at and play around with. But in human growth hormone, there is no glycan there. So we asked the question, could we stick a site in there so that we could put a glycan in there? And why do we want to put it here? We want to put it here because this is the receptor down here. And so this glycan is away from where the receptor interaction would be. So it wouldn't interfere with the interaction of the protein with the receptor, but it would still help with its proteolysis stability and circulating half-life because of the interaction with the acyl-glycoprotein receptor. So our first generation of biosynthetic pathway then involved 2 enzymes that transfer a sugar, an enzyme that converts a blue square into a yellow square because E. coli doesn't make yellow squares. And so this is an epimerase. And then we needed something to help with the formation of disulfide bonds in the cytoplasm of E. coli because that's also not a natural occurrence. And there are commercially available strains of E. coli that do that, but they don't do it particularly well. So we added a gene called the protein disulfide isomerase, and it just helps the 2 cysteine residues find each other and make a correct disulfide bond. So then what we ended up with was our protein of interest with that threonine with the free hydroxyl group in the loop, and then we get this disaccharide structure in the first iteration put onto this protein. So the engineering of that loop is not random. We didn't just sit down and say, okay, what are all the permutations we could think of? We actually had some guidance from a really nice piece of software that was developed at University of Texas at El Paso by a fellow by the name of Thomas Gherkin. And he and his colleagues have for a number of years been collecting data on what does a glycosylation site look like in a protein that carries one of these glycans. And they compiled all of that information and then developed an algorithm to predict if you put a sequence in, what would the probability be of it being used by a particular enzyme that actually puts that first sugar on? And so they call that isoglyP. And what we were able to do was to take the native interferon alpha-2b amino acid sequence, which looked like this, and we swapped out the VGV for a PQP, and we got about a 4-fold increase based on the software. And I'm going to show you in a minute that we get quite a large increase in the ability of that particular amino acid to be modified. We did the same thing with human growth hormone, and as I told you, this sequence isn't normally glycosylated. It's got a very low probability, and we looked at 2 versions, and this second one actually works pretty well. So if we just look then at the quantification of protein, so we grew up this strain that now has a plasmid expressing our target protein, and then we've got, in this case, 2 plasmids. And the second plasmid actually encoding that biochemical pathway that I told you about for making the modified sugar that we need. And then we just stack up the amount of unmodified protein, the amount with 1 sugar and the amount with 2, because remember, this is our first iteration, so the final glycan's only 2 sugars in this experiment. And what we found was that with the native sequence, if we use 2 different E. coli strains, you can see that at best we're getting something like less than 40% modification, and in this strain quite a bit less. These are just 2 different E. coli strains that are supposedly optimized for that disulfide bond formation. However, if we look at this modified sequon, we now go from about 40% to about 75%, which is pretty good for just a very modest change in the sequence. And now actually both strains are working quite a bit better than they were just by the 3 amino acid change that we made. So we moved on from that first generation to a second generation where now we've got all the enzymes we need to make that tetrasaccharide, so put all 4 sugars on. And we've got the 2 transferases, that enzyme that converts 1 sugar into the other so that we can put the square on in the first, the disulfide bond isomerase, and then 2 other transferases to round out that tetrasaccharide. And we haven't yet quantitated the data from this the way that we did for the first experiment, but now that our new HPLC is here and functioning, we're hoping to be able to do that pretty soon. But what I am going to show you is some mass spectrometry data, which is not quantitative, but it gives a pretty good indication of the different kinds of species that are present in the strain. So if we look at that engineered version of human growth hormone, and we look now at, actually this is a version where there are only 3 transferases present, so the structure that we should, or that we hope to get, is 3. And what we can see is that there's still a substantial amount of unmodified protein here and a little bit of just the starting material. And we've got pretty good production, but it's not great. And then if we go on to the scenario where we've got all 4 enzymes that should make this tetrasaccharide structure, it's kind of the same. We've still got quite a bit of unmodified material, and we've got a little bit of the intermediate type structures. However, when we add in an enzyme that takes this starting structure and modifies it with this, when we've got interferon, it actually gets rid of pretty much all of the unmodified protein. So it's just kind of a shift in chemical equilibrium, if you will, right? So you're making product, pulling things toward the product side. There's a little tiny bit of unmodified, and there's a little tiny bit of just the 1 sugar here, and the same thing is true over here. We have a little bit of just the 3 sugar version, but I think it's coming along quite nicely. And what we're playing with right now is just different growth conditions for these things. And I should mention that one of the things that we have to be able to do is make sure that this is reproducible. So from culture to culture, from day to day, and anybody who's ever worked with E. coli will know that from day to day, there's no guarantee that it's going to be exactly the same growth as it was the day before, which is unfortunate, but we sort of round that out by doing statistics, if you will. So we do enough growth so that on average, we're happy with how this yield is coming out. Okay, so we've worked with a lot of companies, and companies always are quick to point out that if they've produced the native protein and they've done all of their clinical testing on the native protein, you just change some amino acids in that protein, it's no longer the same. And in the eyes of the FDA, it's a new chemical entity. And so it needs pretty much full testing, although they're relaxing the rules a little bit. So basically, what we have is an unnatural structure for human growth hormone for sure, because there's no glycan on there naturally anyway. And so it turns out, and I didn't know this before we started, that the biological assay for HGH is not trivial. It's not where you just throw HGH on cells and watch proliferation or something simple like that. It regulates its own production, and most cells produce it, and so when you throw it in, it produces some, and so it gets complicated. And so we had to contact a real expert, and there's a fellow by the name of Andrew Brooks in University of Queensland in Australia who's offered to look, he's an expert in HGH, and he's offered to test our proteins in his system. And he's familiar with HGH made in E. coli because they make that all the time. It has no glycan on it at all. So it's a good control for us. Okay, so our follow-up work then is going to be to extend these strain designs by looking at more target proteins. So I mentioned at the beginning that interferon alpha-2b has this associated toxicity. So we've started looking at something called interferon lambda-3, which has kind of the same mode of action but far less toxicity. And we tried a version of that, interferon interferon lambda-2, which didn't express very well in our hands, but we're confident that 3 will work well, he says. We're going to start looking at putting multiple glycans on, because the more the merrier in terms of keeping the thing in solution. We're going to get into looking at different core types, because these have the potential for higher branching structures, and the more branching that you can have, the better that would be for keeping it out of the clutches of the acyl-glycoprotein receptor. And there are some proteins for which the core 2 glycan is natural, and so we'd like to be able to make the natural structure. And we'd like to get rid of that second plasmid by putting everything into the chromosome. We've done some of that already. We have one biochemical pathway there, but we'd like to get 2 there. But it's been a bit of a trial and error trying to get 2 biochemical pathways inserted into the genome and functioning at the same time. So anyway, we want to do that so that we can decrease the metabolic burden on the cells. All right, so just a couple of acknowledgments then. My longtime collaborator, Steve Withers, has been working on this project for a long time. And Leanne Sim, who was actually a postdoc with me and is now a research associate with Steve, has been driving a lot of the synthetic biology for this project. People in my lab in Toronto who did a lot of work, especially postdoc Ting Du, who's now at the Dana-Farber Cancer Center in Boston. And then the new crew here, Nicole Thompson, my technician, and then 2 undergrads who are plugging along, getting things going. And this is what the Wakarchuk lab looks like at the moment. So this is the Edmonton crew, and this is the existing Toronto crew. And you'll notice that we have a clone who travels back and forth. His name is Alex, and Alex was here a couple weeks ago, so we managed to get his picture while he was here. So thanks for your attention, and I'd love to entertain questions. Warren.
If you want to convert blood types away from those that are recognized by specific antibodies in the individuals. Couldn't you also just add sugars as well? Because that would be a novel sugar that the immune system's never seen before. And,
Yes,
go ahead.
that would also be possible. I'm not sure whether additional sugars mess up the function of red cells. I mean, I guess red cells just circulate and keep hemoglobin, so they're like they don't have a nucleus, they don't divide, they don't do any of those things. So that might be another strategy.
To what extent are these methods likely to be used to divert autoimmune diseases? Is there any work at all in that direction? I know that's a very broad question, but I'm thinking, recognition of, human, myelin sheaths seems sometimes to be triggered by various, maybe bacterial infections or something.
Right. Yeah. I mean, I think there's a lot of interest in trying to use glycobiology to look at those things. So the myelin sheath, for example, is held together with a particular glycolipid that's absolutely essential for the proper deposition of myelin, and it's one of the things that's destroyed in MS, for example. And so if we could look at a way of helping not only to redeposit myelin, but reintroduce that particular glycolipid at the right place, then that might work. One of the things that I didn't get a chance to talk about today is that we have a project on cellular therapies where we're actually thinking about engineering Schwann cells, for example, who are the myelin depositors, so that they can be used under situations where you might want to remyelinate nerves. And we've had some pretty positive experience with that so far. But there are lots of areas where either proteins or cells could be used in the context of those autoimmune diseases. So.
The glycocalyx that's found in insect midgut cells is critical in its ability to take up various pathogens, probably even things like Plasmodium, but certainly viruses that I work with. Do you suppose that you could develop some kind of strategy whereby you could change sort of what's synthesized and deposited on an insect cell?
I think, the short answer to that is yes. So people have been engineering insect cells mainly for protein production, but they've been introducing human glycan pathways. Does CRISPR work in insect cells? Because presumably you could CRISPR out or in something that would change the way, because the basic pathway is exactly the same.
Yes, that was my question. So
Yeah.
if it works in this system, you might be able to. But I don't think insects use as much sialic acid in their.
Well, it's actually controversial as to whether they actually have sialic acid or not, right? I suspect that they have a form of sialic acid that isn't necessarily the regular, what we call Nu5AC. It's probably a relative which doesn't go detected in a sialic acid assay, but still has the same kind of function, right? So sialic acid is a very special sugar with that carboxylic acid function and that glycerol side chain. But there's, if you think about things like pseudominic acid or legionaminic acid, which are basically isomers of that and occur all throughout the bacterial world, and I think in some plants, it would make perfect sense that they probably would be found in insects as well, I suspect. That would be my prediction.
I was wondering if your modified interferon actually, maybe it doesn't act on the hypothalamus to make you feel like crap.
That's a good point.
Regular interferon does.
Yeah, all we've done so far has just been the in vitro tests, and basically just looking at tickling the receptor and then following the phosphorylation cascade that's downstream of that. But that's a good point. And again, that modified interferon would also have to be retested. So, yeah.
So, yeah,
polysaccharides, I mean, there's dual possibilities. One is this highly specific lectin-like interactions where it really matters which sugar and which orientation. But then there's also this idea that a general hydrophilic cloud may be functionally important, and it's not the specific structure, but just the presence of a certain mass of hydrophilic stuff. Do you have any sense of whether that latter case is actually true? And if so, how, do you have any idea of whether the effect you're getting by modifying the interferons is something specifically affecting a very discrete molecular interaction, or just some kind of more general solubility prevention of interaction?
Right.
Sterically?
Yeah, that's a complicated question, but I'll try to give you a less complicated answer. So the extracellular matrix is often thought of as this jumble of extracellular polysaccharides that's kind of has this nonspecific interaction. But if you look at a protein like laminin, it has very discrete binding domains which recognize carbohydrate substructures that have been characterized. So the same thing is true for fibronectin, and there are lots of proteins that interact with hyaluronan and other glycosaminoglycans where they have specificity for a certain number of repeat structures in the case of hyaluronan, or in the case of something like heparin where you have this modified sulfation pattern, they're absolutely specific for a sulfation pattern. So I think in the extracellular matrix, while there is that bulk hydrophilic gel-like quality, which is really important, and it's super important for things like your joint function, right? So, joint compression doesn't work unless you've got glycosaminoglycans to soak up all that water. That's a non-specific hydrophilic function. But I think effectors binding to glycans, that's quite a bit more specific, I think.
Any more questions? I guess I have a naive one, which is this is a $210 billion industry, right? That's what you quoted.
Yeah.
So when you decide to go up an avenue of research here, and big pharma is maybe looking over your shoulder, or they're competing with you, how does that all work? I mean, how do you decide what it is you're going to tackle, and not knowing that 20 other people might be doing the same thing? You've got much deeper pockets,
Right.
maybe south of the border.
Yeah, so the way that we avoid that is by talking to pharma first and saying, if we could do that, would you be interested? And they go, yep, come back when you're ready to talk. So one of the nice things about GlycoNet is that all of those projects were vetted for commercial potential. And so they all went through 2 levels of review, scientific, and then looking people from the pharma industry saying, yeah, that has potential to be of interest to a variety of people or not. And projects that didn't meet muster for the commercial side were still deemed interesting scientifically. Some of those were funded, but these ones where we're actually trying to make a product, actually we had to go to pharma and say, if we build this, are you interested in it? And the answer to, usually they say yes, because it's no problem for them to let somebody else commit funds to fund the work, and then they'll scoop it up if it proves worthy. But we've chatted with 2 or 3 companies who are interested in these small cytokine things, pending testing and some of that stuff.
So you're far enough upstream that you're
yes,
safe.
Yeah.
Okay,
Yeah.
until you get close.
Yes,
Yeah. Is there any other questions? So I want to thank Warren. I'm going to bestow upon you the
Ooh, my very own water bottle.
water bottle.
Awesome.
You have to give one of these talks or earn one of these bottles. So, thanks,
All right,
Warren.
all right. Thanks very much.
And there's still some carbohydrates left here.
Speaking next time.
Oh, I don't remember. Colleen. Thank you, Colleen. I forgot to look that up.
Yeah.
It was on my piece of paper. I think I'm tired.
Oh.